Hello, everyone. In this presentation, we'll explore how scientists verify molecular identity in biologic medicines using peptide sequencing, peptide mapping, and NMR spectroscopy. These techniques play a critical role in sameness studies, especially when developing biosimilars or complex biologics. More importantly, we will see why orthogonal analytical methods are essential for ensuring reliability, regulatory confidence, and ultimately patient safety. Sameness studies are analytical evaluations used to confirm that a biologic product matches a reference product at multiple structural levels. First, scientists verify the primary structure, meaning the exact amino acid sequence of the protein. Next, they analyze post-translational modifications, such as glycosylation or oxidation, which can affect the molecule's function. Finally, they evaluate the higher-order structure, which refers to how the protein folds in 3 dimensions. These studies are especially important in the development of biosimilars and peptide therapeutics. When studying complex biologics, relying on a single analytical method is rarely sufficient. Orthogonal methods use different scientific principles to analyze the same molecule. This approach provides independent verification, helping scientists confirm results from multiple perspectives. It also reduces analytical bias and minimizes the risk of errors or missed structural differences. Because of this, regulatory agencies such as the FDA and EMA strongly encourage the use of orthogonal analytical techniques. Peptide sequencing focuses on confirming the exact order of amino acids in a protein. This is typically performed using liquid chromatography coupled with tandem mass spectrometry, or LC-MS. The protein is first broken into smaller peptide fragments, which are then analyzed to determine their mass and fragmentation patterns. From this data, scientists reconstruct the complete amino acid sequence. This step is essential to ensure that the therapeutic protein matches the intended molecular blueprint. Peptide mapping is often described as creating a molecular fingerprint of the protein. In this method, enzymes digest the protein into predictable fragments. These peptides are then separated and analyzed using liquid chromatography and mass spectrometry. Peptide mapping helps detect post-translational modifications, such as oxidation, deamidation, or glycosylation. Because it provides site-specific information, peptide mapping is a powerful tool for comparing structural details between products. While mass spectrometry focuses on sequence and modifications, NMR spectroscopy examines the protein's 3-dimensional structure. NMR works by observing how atomic nuclei respond to magnetic fields. These signals reflect the chemical environment and spatial arrangement of atoms within the molecule. Because the protein is analyzed in solution, NMR can detect subtle changes in folding or conformation. This makes it an important technique for confirming the structural integrity of biologic molecules. Each of these analytical techniques provides different types of structural information. Peptide sequencing confirms the exact amino acid order and sequence variants. Peptide mapping identifies post-translational modifications and peptide fingerprints across the molecule. And NMR spectroscopy evaluates the higher-order structure and folding patterns. Together, these complementary techniques provide a comprehensive structural characterization of biologic products. In industry laboratories, these techniques are often used in a structured workflow. First, the protein is digested into peptides using specific enzymes. Next, peptide mapping is performed using LC-MS to generate a detailed molecular fingerprint. Sequencing analysis confirms the identity of individual peptide fragments. Finally, NMR spectroscopy analyzes the intact protein structure in solution. All results are then compared with reference data to confirm product similarity. Regulatory agencies require rigorous analytical characterization of biologic products. Guidelines such as ICH Q6b emphasize detailed evaluation of structure at multiple levels. Both the FDA and EMA recommend using multiple orthogonal analytical techniques as part of the totality of evidence approach. This strategy reduces the risk that subtle structural differences might be overlooked. Ultimately, these requirements help ensure consistent product quality and patient safety. To summarize, confirming molecular sameness requires multiple layers of structural analysis. Peptide sequencing verifies the correct amino acid sequence. Peptide mapping provides detailed insights into post-translational modifications. And NMR spectroscopy evaluates the three-dimensional structure and stability of the molecule. Together, these orthogonal methods create a strong analytical framework. As the quote suggests, orthogonality is not just useful, it is fundamental to trust and safety in modern biologic development.